Welcome back. In this session, we are going to discuss the a very important lifestyle disease, which is called hypertension. A lifestyle disease I say because this the major reason for hypertension is the stress that is associated and then what is the chemistry that goes on which elevates the blood pressure. Okay. Now, there are many mechanisms. Before I go into the details, I can say that there are many mechanisms by which the blood pressure can be elevated. One simple example, if somebody takes lot of salt in his diet, sodium chloride, then the amount of sodium in the blood is high, and that is one way to increase the blood pressure. Okay. And there are also calcium, movement of calcium through the channels, that is also an important component of mechanism of blood pressure, mechanism of hiring the blood pressure, okay, or increasing the blood pressure. So, you can have calcium channel blockers to do for the lowering of the blood pressure. There are other many methods of lowering the hypertension. Another thing which is very common for higher blood pressure is the narrowing of the arteries. That means, the arteries through which the blood flows, if the artery dimension, the diameter becomes less, then what will happen, that the same amount of blood, to push same amount of blood through that narrow passage, the pressure goes high. Okay. And the whole thing is narrowed down because of the deposition of mainly cholesterol type substances or cholesterol, and that is what is called atherosclerosis, where there is this deposition of cholesterol within the walls of the artery and narrowing the artery. So, that is another way, means reason for how. So, higher cholesterol means at some point of time you are going to have this deposition of cholesterol, and that will also a causative agent of high blood pressure. But today we are not discussing all these three. What we are discussing is another way, which is called humoral way of elevating blood pressure. Humoral means the body fluid, humoral mechanism of hypertension. Humoral means through body fluid, that how can you increase the blood pressure, okay. Like sodium, if the body fluid means the blood contains more sodium, then you will have higher blood pressure. Okay. Now, sodium is a little bit easier because you can cut down the intake of the salt, but if there are breakdown of other metabolic processes, then that you have to be very careful. You can target those metabolic processes and then see how the blood pressure can be controlled. Okay. Now, one such humoral mechanism of blood pressure, increasing the blood pressure is what is called the Renin-Angiotensin system. I will come back to that Renin-Angiotensin system. Before that, just what are the hypertension, how this blood pressure is commonly known, that if some people say that I am suffering from hypertension. Hypertension means is the pressure, hyper means high. So, higher tension means higher blood pressure, this is also abbreviated as HTN or even just higher tension, also known as high blood pressure HBP. If it is a long term medical condition in which blood pressure of the artery is persistently elevated. High blood pressure typically does not cause symptoms, but long term high blood pressure is a major risk factor for coronary artery diseases, because arteries get it is found that blood pressure has a connection with higher cholesterol level also. So, your arteries get narrower, and whatever inside the cholesterol crust is there is a layer of cholesterol which is solid. So, if that detaches from the inside of the artery, so that ultimately flows through the blood and blocks your heart valve or it can go to the brain and cause the cause what is known as the cerebral attack. Okay. So, it is a major risk factor of coronary artery disease, stroke, heart failure, vision loss, chronic kidney disease and dementia, because there are cases when this high blood pressure goes, when the people suffering from high blood pressure, their brain function also slowly decreases, and then ultimately it will cause what is called dementia. That means, you forget things. Kidney disease is another one, because of the high blood pressure, kidney faces lot of trouble in flushing out the unwanted things, and so the long term effect is a chronic kidney disease. Now, I told you that I will be talking about the renin-angiotensin system. The renin-angiotensin system is one of the humoral mechanisms of blood pressure control. It was known about 60 years ago that what is the biochemistry of renin-angiotensin system. Now, I will tell you what is in the next slide. Before I read this whatever is written here, I show you a schematic diagram. Okay. In the body, there is a protein which is called angiotensinogen, which is a bigger protein. Okay. This angiotensinogen is hydrolyzed by an enzyme which is called renin, and when it is hydrolyzed, it makes a smaller peptide that contains 10 amino acids. So, angiotensinogen, this word means gen, like pepsinogen, chymotrypsinogen means a precursor of the actual compound. So, chymotrypsinogen is the precursor of chymotrypsin. Okay. Then pepsinogen is the precursor of pepsin. Similarly, angiotensinogen is the precursor of angiotensin, but this angiotensin is called angiotensin 1. That means, this decapeptide 10 amino acid containing peptide is obtained from angiotensinogen by action of the renin. So, what is renin? Renin must be a protease, because it is hydrolyzing this big protein into a very small peptide 10. So, this is angiotensin 1. Then angiotensin is converted into angiotensin 2. Angiotensin 2 is 8 amino acid containing peptide, 8 amino acid containing peptide. So, that means, now this is what is called angiotensin converting enzyme, the enzyme that is here, which is also a protease by the way, because you are removing amino acids by hydrolyzing the peptide bond. So, that is called angiotensin converting enzyme, ACE. Okay. Angiotensin converting enzyme. Now, this angiotensin 2, which is obtained from angiotensin 1, is a 8 amino acid containing peptide. Now, it is like basically a hormone. It has got receptors, and it binds to the receptors, and then it has its own receptors. It binds there, and what is the effect? What is the signal that it creates? When it binds to the receptor, it causes vasoconstriction. Vasoconstriction means your blood vessels become narrower. It is not by any deposit, it is by the action of this hormone angiotensin 2. So, the blood vessel contracts. Okay. So, that is what is called vasoconstriction. So, if there is vasoconstriction, that means the blood pressure is going to be increase. Okay. So, that means angiotensin 2 binds to the receptor and sends a signal which causes vasoconstriction. So, there is elevation of blood pressure. So, angiotensin 2 has been implicated in the elevation of blood pressure. But in addition to that, there are other there are many harmful effects of angiotensin 2. One is that there is a protein or a small peptide which is called bradykinin. That is another hormone, bradykinin, but the bradykinin is just the opposite of your angiotensin 2. Bradykinin is a vasodilator. Vasodilator means it increases the area of the volume, that diameter of the blood vessel. So, if that is the case, that means you get vasodilation. Vasodilation means lowering of blood pressure, okay, and vasoconstriction means elevation of blood pressure. So, via vasoconstriction this elevates the blood pressure, because angiotensin 2 works in that fashion. It binds to the receptor, causing the elevation of blood pressure. The other important thing is that this angiotensin converting enzyme, which hydrolyzes angiotensin 2 and makes angiotensin 1 and making angiotensin 2, it also breaks down, it also hydrolyzes bradykinin and destroys it. So, if it destroys the bradykinin, that means your vasodilation activity by bradykinin, that also is lowered. So, that means this combined effect will even further increase the blood pressure, okay. And that is not the end of it. This angiotensin 2, when it binds to a receptor, it not only does vasoconstriction, but it also sends signal which then sends a signal which ultimately leads to the release of another hormone which is called aldosterone or aldosterone, that is a steroid. A steroidal hormone which is called aldosterone. Now, if it is a hormone, then it has got its own receptor. So, aldosterone now binds to its receptor, and the signal that it sends is that do not lose the sodium, better retain the sodium and remove the or excrete the potassium, okay. And also you retain the water, okay. So, all these, that means there are 4 kinds of effects. One is just direct vasoconstriction by angiotensin 2. Another is this enzyme, angiotensin converting enzyme, destroys bradykinin, which is a vasodilator. And third, this it also enhances the release of another steroidal hormone, that is called aldosterone, and this aldosterone enhances the retention of sodium and water in the blood. So, all these combined together has a disastrous effect on elevation of blood pressure. Okay. So, that is the chemistry humoral mechanism of how the renin-angiotensin system works. Okay. Now, you can say that if I want to have an anti-hypertensive drugs now, so my best, what will be my best bet? I can use inhibitor of angiotensin converting enzyme, because if I inhibit angiotensin converting enzyme, that means that inhibits the production of angiotensin 2, that inhibits the breakdown of bradykinin, that if angiotensin 2 is not produced, that means vasoconstriction will be less, and that means the release of aldosterone will be less, and release of aldosterone less means less retention of sodium and water. Okay. So, you can target angiotensin converting enzyme. You can say that I can actually target renin also. Renin is the first enzyme in the process, and then I can target renin and then stop angiotensin 1 from producing. If angiotensin is not 1 is not there, then obviously, even if the SEEE, that means the angiotensin converting enzyme is active, but your lack of substrate concentration will tell you that angiotensin 2 will be reduced, will be produced in less amount. So, renin also is a target, and angiotensin converting enzyme is also a target. Now, angiotensin, now let us consider these two these enzymes but we will not talk about the renin, but obviously renin is also a target, but most of the successes that have come is basically the inhibition of the angiotensin converting enzyme. That is the major research that was done during the 80s, and then the first set of drugs based on ACE inhibitors came into the market, and today they are also sold in large quantity in the market. But beside that, there is another one which we should not overlook, that is angiotensin 2 has receptors, right, has see it when it exerts vasoconstriction, that means it binds to the receptor and then sends the signal which creates vasoconstriction. So, what you can do, you can, if you can make a receptor antagonist of angiotensin 2, then what will happen? Even if angiotensin is produced in large amount. So, this receptor, the receptor, if you are using an external agent, remember, which is an antagonist. Antagonist means it binds here and does not produce any effect. Okay. So, the angiotensin 2 cannot bind, and so the vasoconstriction effect of angiotensin 2 is gone, and if you can block the other receptor which also sends the release of the aldosterone, then that will also, but you have to use antagonist, remember antagonist, so that this part can be taken off. Although this ACE degrading bradykinin, ACE means the enzyme, you are not inhibiting the enzyme, what you are doing? You are inhibiting the receptor sites utilizing antagonist. So, there are two kinds of drugs based on this angiotensin renin-angiotensin system. One is ACE inhibitors and the other is ACE receptor, not ACE, sorry, this is the angiotensin 2 I can just simply angio 2 receptor antagonist. Okay. So, these are the two strategies you can take. We will talk about in this medicinal chemistry section, we will talk about the ACE inhibitors, how they are designed and how the companies ultimately were able to find drugs and which are occupying a large section of the anti-hypertensive drugs today in the market. Okay. Now, let us these are the, I have not said what are the amino acid sequences of this angiotensinogen or what is angiotensin 1. Some partial sequence of angiotensinogen is shown here. Okay, you do not have to remember this. So, this is a big protein. So, when renin works, first enzyme is renin, angiotensin is converted into angiotensin 1, which is a decapeptide, that means 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, this renin actually cleaves the bond between leucine and valine. Okay. It cleaves the bond between leucine and valine and then forms this decapeptide which is called angiotensin 1. Then angiotensin converting enzyme comes and converts the 1 into 2. So, what it does? It breaks now the bond between phenylalanine and histidine and removes the dipeptide, and this is the octapeptide which is the angiotensin 2. Okay. So, this is our compound, we are mostly concerned with this compound and we are mostly concerned with this enzyme, because we have seen the disastrous effect that these things can have. However, just to extrapolate it a little bit, see angiotensin 2 is also can be degraded further by an aminopeptidase. Aminopeptidase means which cleaves from the amine side, the nitrogen side. So, aminopeptidase takes the aspartate off and forms what is called angiotensin 3. and another aminopeptidase N which cleaves the dipeptide from the N terminus, and that means it is a 5 1 2 3 4 5 6, it is a 6 amino acid containing one, and here it is 7 1 2 3 4 5 6 7. So, this is angiotensin 3, this is angiotensin 4. Now, the question is, if you stop the production of angiotensin 2 by ACE, that will also lower the production of angiotensin 4 and angiotensin 3. The question is whether they have any beneficial effect, or if they are also part of this blood pressure elevating process. Interestingly, what has been found that angiotensin 3 and angiotensin 4, they are also responsible partly for the elevation of the blood pressure. So, that is good that if you can stop the production of ACE, the angiotensin 2, that means you lower the concentration of angiotensin 3, you lower the concentration of angiotensin 4. So, basically with one strategy you can stop the production of so many blood pressure elevating agents. Okay. Now, let us see what type of enzyme angiotensin, this ACE. ACE takes out the dipeptide from the C terminus. Okay. Now, you know, if something is hydrolyzing from the C terminus, that is called carboxypeptidase. Okay. But carboxypeptidase is usually the breakage of the last amide bond, like carboxypeptidase A, which takes up all amino acids except the basic amino acids here, carboxypeptidase A. So, this is called a dicarboxypeptidase A, because it actually hydrolyzes not the last one, but the penultimate one, because it is releasing a dipeptide. Okay. So, carboxypeptidase A is breaks here, that means there is one particular amino acid here, but this dicarboxypeptidase is basically breaking the penultimate. Penultimate means not the last one, before that the penultimate, like ultimate day of World Cup cricket final is tomorrow. So, today is the penultimate day for the World Cup final. Okay. So, that is the penultimate amide bond A1 A2. Okay. So, it is a dicarboxypeptidase. Now, if you want to make an inhibitor of angiotensin converting enzyme, you need to know what I told you in the drug design, the initial introductory part, that for drug design process you need to know the target. The target is known, target is angiotensin converting enzyme, but then it is better that you note the structure of this compound, that means the crystal structure, the 3-dimensional structure of it. Okay. Because angiotensin converting enzyme, remember, is not the protein, is not the small peptide, it makes the small peptide angiotensin 2, but angiotensin converting enzyme is an enzyme. So, the question is, when this research started, whether angiotensin converting enzyme structure was known or not. It was not known at that time. Okay. It was not known at the time, but what they found that it is there is a metal involved in the breakdown of this peptide bond, and there is the enzyme, the well known enzyme whose crystal structure was known those days was carboxypeptidase A. So, carboxypeptidase, it was known that it is a zinc based enzyme, and what they can do, what they knew at that time, that this is carboxypeptidase, I can write C peptidase A, and this ACE, angiotensin converting enzyme, they found that this is also a zinc based dicarboxypeptidase. Okay. So, the first assumption is that this, as the scientist, I believe it is in Merck Laboratories or some big pharmaceutical company, they started this research, and they first assumed that the carboxypeptidase A has the structure, because its crystal structure was known, the structure of ACE is very similar, it resembles the carboxypeptidase A. So, they took carboxypeptidase as a model for designing the intended inhibitor for ACE. Okay. So, that was the starting point. Now, you can say that if this was wrong, then what would have been the case, but there are analogies because it is a zinc based enzyme, that is also a zinc based enzyme. Okay. So, that was the major analogy. So, it must be having a similar mechanism like the carboxypeptidase A. Now, what is carboxypeptidase A? It has got an amino acid, it has got an amino acid CO2 minus, and it accepts actually except the basic amino acids, it accepts other amino acids like phenylalanine. Say phenylalanine, carboxylic acid, and then phenylalanine is connected via a peptide bond to other amino acids. Okay. So, now the carboxypeptidase is hydrolyzing this bond, that we know, and it is a metalloenzymes. So, what is the function of the metal? That is number one, function of the metal. That is one question. The second question is what could be the active site pockets, Since it is a carboxypeptidase and we know that the C terminus ends with a CO2 minus. So, definitely what we can say, roughly we can predict, even if we do not know the extra crystal structure, or see what it can happen, that at the end there must be some pocket which is having a positive charge like NH3 plus, because if that be the case, then only the CO2 minus can go and bind here. Similarly, you can say for amine terminus, hydrolysis at the amine terminus, the amine is usually present as NH3 plus. So, the binding takes place at a CO2 minus. So, there must be here the plus, so that they can form a salt bridge or electrostatic interaction. So, then you have suppose a phenylalanine, say CH2PH, and then you have a CO, NH, you have a CO. Actually, I should not write this way, because the NH is the anti side. Okay. So, maybe we write NH here. Sorry, what is happening? So, that is NH, that is CO, and then other amino acids. So, I can say definitely there must be a zinc here which is attached, because it is zinc mediated hydrolysis. So, the carbonyl is having this the zinc plus 2 is acting as a Lewis acid. So, the carbonyl oxygen is chelated to the zinc, which activates the oxygen, and then ultimately in turn that activates the carbon for attacking by a nucleophile. Okay. And this phenyl which is there must be some pocket here which is hydrophobic pocket that stabilizes the phenyl. So, I can, without going into any details, I can say that this is the situation for carboxypeptidase. Okay. So, for ACE, then what will happen? Now, ACE, remember that ACE has a it hydrolyzes the dipeptide, that means the penultimate peptide bond. So, it will have, so definitely there is a CO2 minus, and then this whatever amino acid it is here, R, suppose R1, then you have NH, then you have CO, but the zinc must not be here, because if zinc is here, then the terminal peptide bond would have been hydrolyzed. So, then there is some compound here, suppose this is R2, and then you have this NH, then you have this CO. So, now you can draw a kind of active site structure. So, there must be zinc here, then there must be some group which stabilizes this R2, then there must be some group, but that is not zinc, it could be a hydrogen bond with some, it via hydrogen bond it is stabilized, and then another stabilizing factor here and a plus charge somewhere here, so that the carboxy stabilized. So, you can say based on this, you can say that this could be the rough sketch of the active site of angiotensin converting enzyme. Okay. So, from here we will proceed to the next session, because it will take some time, it cannot be completed at this point. There is a lot of discussion after that, and very interesting chemistry came out of specifically the design strategy came out of this angiotensin converting enzyme inhibitors. And today when we talk about there is a common terminology, that rational drug design, that some courses are called rational drug design, but I have deliberately excluded that word rational. I have said organic chemistry of biology, in biology and drug development. Okay. I did not say rational drug development, because although the drugs are developed by a logic, the logic is that from comparing with different enzymes, finding a target, which forms the part of the basis of the drug discovery process, but always try to remember that nothing is ab initio. Ab initio means from scratch it will be very difficult to really develop a drug. Like the case of aspirin, I told you that there are some plants, willow tree, you get this salicin, which is an glycoside of salicyl alcohol, and from that ultimately aspirin was discovered. Similarly, there are dihumoral, that we will discuss that, it is a vitamin K, I think we have discussed already dihumoral. Dihumoral, that is also a serendipitous discovery from something people observed, and then they try to modify whatever the observation is. So, ab initio drug discovery is almost impossible. There must be some clue somewhere from where we can build up our or build on our rationale to design the drug. So, a lot of people criticize that rational drug discovery may not be appropriate, because you already have some preliminary knowledge of something, and then from there you start discovering the drugs. But anyway, that term I do not mind keeping the rational word. What I mind is that if you say ab initio drug discovery, that is a big problem. Ab initio drug discovery means from scratch without knowing anything, that is very difficult, that is usually not possible. Okay. Some type of clue or hint has to be there in drug discovery. Okay. So, the next session we will come back to this aspect again. Thank you.